Auzubillahi minash shaitani rajim. Bismillahir Rahmanir Rahim. Dear respected chairs of the 13th Annual Quran and Science Symposium, respected guests, and the attendees of the MSLM 26 conference as a whole, Assalamu alaikum wa rahmatullahi wa barakatuhu. The topic of my presentation today is regulatory T cells as peacemakers of immune balance. The beautiful recitation of the Surah Ar-Rahman that we heard at the start of this session, verses 7 to 9, draw our attention to the principle of mizan or balance. These verses highlight that the universe operates according to the laws established by Almighty Allah. From the vast celestial bodies in the heavens to the smallest plants growing upon the earth, all creation performs its allotted function with remarkable regularity and precision. The solar system and the components of the universe work together in perfect harmony. And this harmony depends upon balance. And even the slightest disturbance of this harmony or deviation from the order established by Allah could disrupt the equilibrium of the entire system. The Holy Quran draws our attention to the profound principle here. It is a universal principle. The stability and flourishing of a creation are sustained through balance, order, and regulation. And this draws our attention to this very biological principle existing in the science of the immune system. It's the same equilibrium that the immune system is operating and needs to be balanced at all times. It is a delicate equilibrium, strong enough to defend against infection, yet restrained enough to prevent damage to cell. And that opens the topic of T regulatory cells and their role in maintaining this balance. So here is what we can relate about the immune system to the verses that we just recited. And as I was reflecting on these verses, it's amazing how the guidance from the Holy Quran about reflecting on the particular verses that drive scientific inquiry is well illustrated in the biology of the immune system. And basically, 3 points emerge from this, which is that the creation is governed by the universal phenomena of balance. And if there is a deviation from this balance, even the immune system that is operating has this balance and can be distorted and can lead to adverse circumstances. And I'll go more into the depth of what happens when an immune response gets dysregulated. And the key or the central mediators of this balance are the T regulatory cells. When we look into the history of the regulatory cells, we can just trace back to the mechanism of the immune system. So under normal conditions, when we are in homeostasis, there are two arms of the immune system, the T effector cells, which are the conventional T cells, and they maintain whenever there is an attack on the body or the body gets a pathogen, they defend the body and by mounting an immune response. And once the virus or the pathogen is destroyed, then the immune response tapers down. But there are special cells that are responsible for tapering of this response. And so on the left side of the slide, what you see is homeostasis. Both of those arms are balanced. But sometimes the immune response that is triggered continues to prevail, and it doesn't taper down. And this happens under disease scenario. And this disease scenario is usually the inflammatory or autoimmunity, and you see the dysregulated arm. The cells that are dysregulated are the T regulatory cells. So there is a defect in them. They cannot bring the resolution back to homeostasis. And this slide actually gives a historical perspective. And the reason I'm bringing this up is one of the reasons of presenting this topic was when Dr. Shnaz asked me that, this time the Nobel Prize was for medicine and or physiology was for the 3 scientists who discovered the T regulatory cells and their function. And that's the reason why this discovery perspective is very interesting. And as you can see from this slide, the concept of suppression was there for the immune system. And scientists were thinking about this, that if this system is once activated, what causes it to taper down, right? And so that it's not attacking the own self antigens continuously. So in 1970s, there was this hypothesis of T suppressor arm existing. There's some cells present in the body. But then they could not find a definitive lineage of these cells or definitive markers, and so that hypothesis just died down. And people thought that it was even controversial to think about it. And in 1995, it was Shimon Sakaguchi who found that when he took out these cells out of the mice, took out the T cells from the mice, then they developed strong autoimmunity. And that was the time when these regulatory T cells were discovered, and then their subsequent establishment of function changed the paradigm of modern immunology from just being a phase of immune activation to more a phase of immune regulation. And here is illustrated a brief timeline. I wouldn't say as brief that is shown in the slide, but it has been through a couple of decades when this concept was rationalized, and as early as the 2000s, there was 1970s, there was a concept of the T regulatory cells, but not well defined. And eventually in 1995, the discovery came into limelight, and it was considered a breakthrough. And the reason why it was considered a breakthrough was because of 2 reasons. At the same time, there was 2 other scientists that were working. One was Fred Ramsdell, and the other was Mary Branco. And they were working on establishing the lineage of these cells to see if they were belonging to a characterization that could be defined for their function. And they discovered FOXP3. And one thing I would like the audience to go as a key takeaway is when you talk of suppression of immune response, it is the T regulatory cells and the FOXP3. That defines their function. It's the master regulator, and it's called the fork head transcription factor 3. And these scientists discovered that the same gene of FOXP3 tracked both in mice as well as in human. And because of these core scientific discoveries, the impact was huge. And you know, the impact was phenomenal. They changed the paradigm in immunology, and that paradigm was the immune balance and self-tolerance. And this is just some of the evidences that I mentioned, both from the mice as well as from the human. So in mice, the mutations in the FOXP3 gene will lead to chronic autoimmunity, and these are the scurfy mice. And in human, it leads to an IPEX syndrome, and this is chronic or systemic autoimmunity, and it's called as immune dysregulation and endocrinopathology and entropathy. So this is the concept of the Treg discovery. And just moving into the function of Tregs, I won't go into a lot of depth, is that they produce factors that will inhibit the T effector responses and does maintain the function. This is kind of a schematic that illustrates how they perform their balancing act. They inhibit the T effector cells and then release molecules that will continue to contribute to homeostasis. And this is, again, the same thing that I mentioned about T activation and T cell regulation. The only caveat is when this arm is dysregulated, all these molecular mechanisms get reflected in clinically as a disease, which is autoimmunity. And the current field is trending towards making Treg modulated therapies, and that's the emerging therapies for autoimmune disease, because the reason is they will ensure durable response and durable efficacy in these patients. These were the scientists whose hard work and innovation paid off, and they were awarded the Nobel Prize. And you see how they fall into the perspective of the discovery and the milestones. So I would like to close my talk with a very powerful closing reflection. And this is about not just the discovery of T regulatory cells, but the whole concept here is reading the verses of the Holy Quran and thinking about scientific inquiries if you're a scientist, right? And there could be a broad connection on what the Holy Quran is pointing us towards. And if we analyze that with an innovative mindset, we may uncover truths that could help us to uncover further biological paradigms of life. Jazakallah.